today I want to talk about learning representation of chemical structure and their interpretability. So I was not exactly sure, how much do you know about chemical structure. So I will start kind of talking a bit some introductory topics in learning the chemical structure. So first I want to note that this is a topic that kind of really grows in popularity. I looked just in 2 conferences, ICLEA and NeurIPS, and in 2015 we did the search. There was like no papers that talk about learning molecular representation, and you can see that the number of papers in this area continue to grow. I didn't even include ICML, but there is really more and more papers, and there was recently the whole NeurIPS workshop, I think last week. So lots of activity in this space. And if you are roughly speaking looking at what's going on, there are kind of 4 big areas. So the first one is property prediction. When you are given a molecule and you try to predict some bioactivity or some chemical property. The second one is reaction prediction. It's what we did in high school who studied chemistry. You take 2 molecules and you try to predict what will be the outcome of the reaction. The third one is modeling jointly biology and chemistry. And the final one is doing, what human experts are doing, which is lead optimization or molecular optimization. When you say I have a structure and I want to create another structure which have this improved properties, however we define these properties. So in my talk today, I'm going to talk about interpretability in 2 different ways, and I'm going to touch upon 2 applications: property prediction and generation. So again, in the property prediction, you're given a structure and you want to predict its activity. And it's typically used, this capacity used across all the pharmaceutical companies in 2 ways. The first way, which is kind of traditional way for virtual screening, where you're given, the whole universe of possible molecular structure, you cannot manufacture them all. You want to tell you which one of them are more likely to be promising. So you want to screen them, let's say you're doing anti-cancerous drug, you want to say which one of these molecules are likely to have proper inhibitory activity. And the second way, which is actually more novel, is for de novo design. Whenever you have a generative model which will produce your new molecule, you need to have some guidance to say what is a good structure. So the property predictors are used in these models also, kind of as a guide to find the right structures. So typically, whenever you are thinking of property prediction, you can think about it without any biology. You are given, a structure, and you are given a number, and this number, captures the properties that you care about. So machine sees the structure, the number, and then it tries to learn the mapping between the structure and this number, whatever this number, mysterious number is. So in a very intuitive way, you're trying to do what, medicinal chemistry a long time ago is to say, we know that certain, properties as kind of patterns within a structure correspond to property, let machine learn it. And for a very long time, this is kind of an old idea, decades long idea. People did it using molecular fingerprints. So the way they will do it, they will take a molecule and summarize it as a vector. And this vector would, like a feature vector, it would record what kind of substructures, for instance, this is one way to do it, what kind of substructures do you have in molecule? And given this vector, you can run, any machine learning model. Originally they start with a very simple linear model, then they move to much more advanced models. Then you can predict the property. So the kind of significant change in the technology that happened maybe 5 or 6 years ago was that people realized that you lose a lot of power when you are taking a molecule and then you try to summarize it by these substructures, because it's all driven by human intuition. As we know, it's not a great intuition many times. So the next question was how you can take a molecule and summarize it as a vector in such a way that you can do, the best possible predictions from this vector. And what you're going to lose here, you lose interpretability in some way, because, you know, before you knew that maybe coordinate 1 correspond to this type of ring. Now I don't know what coordinate 1 means. We'll come back to this point later. But this is a very standard way. And, you know, what you hope to achieve when you're doing this kind of translation is that you're going to take let's say we're talking about representation for a particular property, for a specific property. So what you hope to see that the molecules who have similar values of this property, their embeddings, their vectors are close to each other. And of course, molecules which have difference in property, they live in different parts of the space. This is the hope. And if you're very kind of optimistic, that how you can draw it, you can say, I want in this new space that I constructed, the distance in this molecular embedding space, you know, be proportional to the distance in the property value space. It doesn't really happen this way, but at least, you know, it's something that we would like to have. Okay, and you know, a while ago we built this model which is called Chemprop. It's publicly available with you know, people who did it, my students, and many other chemists who contributed the data sets. We ran literally 850 experiments. The model that we have at the time, which was a year ago, was you know, doing fairly well, and it's used in many pharmaceutical companies for virtual screening. Okay, and let me just tell you in few words, you know, how do you build this mysterious vector? So for many of you who are familiar with graph convolution, there is no mystery here. You start with an atom, which would be represented with its own vector. And if you want, if you are a medicinal chemist and you want to put, you know, your own kind of bias, this is a place where you can do it. You can decide what do you put in a vector, in initial vector, which represents each atom. So you can, you know, obviously represent the type of atom, the degree, whether it is in a ring, whatever you like. So after this first step, every atom would have its own vector, but of course it's not a very good representation, because you cannot really you know, do much since you're losing the structure. So then what happens is you start do message passing. So there will be your own vector, like in this case, vector V, and then there will be all these neighbors surrounding it, which have their own, their vectors. And you find a way how to combine this vector into a new representation for vector V, optimizing our objective, which is to improve the property prediction. So after one way of this message passing, you would know about your immediate neighborhood, but since everybody are doing this update at the same time, after the first one hop update, you would eventually get 2 hop updates, because next time when you update with your neighbors, you already know about the broader neighborhood, and you continue. Whenever you are done, you do this number of steps, and you just, every atom would have this new vector, which contains information about itself and some filtered information about the neighborhood. You sum them up. This is a representation for the molecule. And as I said earlier, it actually works pretty well. So Christoph, or maybe I gave him the title, somebody gave the title, which talks about antibiotics. So using this type of model, we had a paper in Cell. So for those of you who don't know, like myself, a computer scientist, Cell is actually a pretty good biology journal. So we got this paper about you know, designing an antibiotic halicin using this model. This was pretty much applying this model. And the paper was, of course, about like biological investigation, but we found really new antibiotics that works very well across multiple drug-resistant bacteria. So, you know, it's kind of useful. And we were very happy. So this paper came in February, and then, you know, the COVID happened, and we were like very enthusiastic of trying to apply this model to try antivirals for COVID. And one thing that we very soon discovered that, you know, the screening data, you know, it was a challenge to get the screening data, but even if you do get the screening data, the main problem with it, that, you know, the screens don't agree with each other. It's small, it has a lot of different problems. It doesn't generalize to different spaces, and the screen is small chemical space. So at that point, we start thinking it's a very naive assumption to think that you can really learn biology, especially if you don't have enough data, using not incorporating any biological information, that you just look just at structures. Now, again, if you have a lot of screening data, you would learn the pattern. But if you don't have enough data, like in case of COVID antivirals, you are in trouble. So what we've done here, we start thinking, how can you change the models? And that's where we're getting to interpretability piece. How can we change the model to incorporate biological information? And luckily enough, there were quite a number of papers in Nature which demonstrate, which find information about different targets that are relevant for COVID. And we used a paper by Gordon where we identify a number of known targets. So what you now want to do is kind of slightly modify your model. You remember before we went from molecule to activity. So now we are going to have this layer in between, which gives you the target. So you're going to go from the structure to the target, and from the target, you're going to go to the activity. And you can see here on the screen, I would not even try to read it, different type of targets that we incorporated. And from these targets, we're trying to predict the final activity. So what was interesting here, that when we started doing this model, it sounded very good, but like all this model that sound good, they typically don't work well. So it didn't work. When we did it this way, like on this picture on the previous slide, it didn't work at all. And it was even worse than not using any targets. And what we realized that one of the key problems was that the target information that we have, that everybody have, is very limited. You never know actually all the targets. And one simple solution that you can address it in this particular layer, when we edit the targets, you can increase the capacity of the model. You can say, being humble and saying, okay, I know these targets, but there is a whole bunch of these latent targets that I don't know what they are. And I am going to be, they don't have a name. I don't know what they biologically mean, but we're just increasing this vector and adding these extra latent variables. And it actually worked very, very well. And we can, as I will show to you, it actually does improve the accuracy of the model quite a bit. So how you can train the model, you have separate data sets which explicitly state drug-target interactions. So you learn the connection between the chemical structure to the targets, and then from the targets you predict bioactivity. So, and you can train it on a single compound data. So what was nice, I'm not going to get into the details, but I have a reference on the next slide, that you can actually take this model, because it explicitly models the biological mechanism. You can actually easily apply them to modeling combinations. And you don't require a lot of screening data for combinations, because you typically don't have it. But you can, through the mechanistic knowledge, to train it to identify how synergistics are combinations of the compounds. And here you can see the reference to this paper. And of course, there is no surprise that once you add these biological targets and you give this extra capacity with latent variables, you actually can do better in predicting activity. And the final thing that I briefly want to say, that's an area where we are working hard now, is how you can look back. So now you have these extra targets that will lead on some molecules, don't lead on another molecules. How can you actually interpret them? And what you can do, because you have screen data for many, many targets in Campbell, which is a data set which is publicly available. In the paper, we go into some detail how you can translate these latent variables into a specific targets. And we were able to discover some new targets that were published in an additional paper by Riva et al. But this is a big area. How do you do translation between, the latent space and observed space? And I don't think that we quite do it right, but that's where we're moving right now. And since I have another few minutes, I want to talk about another task, generation task. So I finished talking to you about prediction, and there, when I was thinking about interpretability, I was thinking about connecting the chemical structure to the biological targets. Now I'm going to talk about interpretability in a different way and for a different task. So the task that people like really love to talk about in the pharmaceutical industry is like generative task. So you can say, I start with one molecule, I have a property predictor that, let's say, tells me how potent is this molecule against some pathogen, and they want to generate a new molecule which will have much stronger activity. So the way you can do it, and those of you who are familiar with autoencoder, it's very direct thought. That's how people started working in this area. You take molecule, you map it into the hidden, into the latent space. You do optimization in the latent space because it's very supposedly small. You can optimize in the latent space, and then you use your decoder to generate a new molecule. So this is another picture here that illustrates this particular thought. Again, you start with some point in the hidden space, you do optimization with your property predictor, find another point, and then generate. And there is a whole bunch of papers that realize this idea using different algorithms. So the first bad piece of news about this paper, that this picture is pretty fake. In a sense that what we discovered and others, that the space is very far from smooth. It's really not a nice terrain. It's like wishful thinking. So when you optimize, you can really get some funny things. But even if you somehow solve that problem, there is a bigger problem here. The bigger problem is that typically when you are working in the pharmaceutical industry, you don't want just to optimize one thing. You care about potency, you care about toxicity, you care about solubility. You have the whole, but there are lots and lots of criteria that you want to optimize at the same time. You may have never even seen any molecules which hold all these desired properties together. So the question is how you can realize this diverse, distribution of property even without ever seeing any examples of these desired molecules. Okay, so this is a challenge. And the way we solved this problem was kind of going back to how traditionally chemists are thinking when they're generating molecule with desired properties. So the way they're doing it, they're taking a molecule and then they say, okay, let's remove this particular subgroup and another subgroup, and you kind of collect the bits and pieces that you believe that when you put them together, they will have the right properties. So this is kind of the strategy that we've done here. The first one, and that's where the interpretability piece comes in, is kind of rational extraction. So what you will do for each property that you care about, and in this case, we will have the red property and the blue property, you are going to train your predictor for the property, but you're not only going to predict how potent is a molecule for, let's say, red property, but you're also going to have an extractive rationale. What I mean by that, you're going to identify a sub-piece of molecule, a functional group or combination of functional group, which drive this property. And then you would have something that take these bits and pieces, and we're going to have a collection of red pieces and collection of blue pieces, and you're going to combine them together and generate a molecule which supposedly would have both green and blue properties, okay? So how you can do rational extraction. So the paper that I will show you the reference to does it, with Monte Carlo research. There are lots of different ways how you can extract this rationale, but pretty much what you are trying to do intuitively, you're trying to identify a sub-piece of molecule that if you remove it, then your property value going to drop significantly. And again, Monte Carlo research is one way. You can do it with reinforced. There are lots of different formulations, but the output of the step would be, your predicted property and this part of substructure that that drives this property. And next, what you are gonna have is you're gonna have again this kind of red pieces, blue pieces. So now you need to learn to expand this subpiece to a complete molecules, and also to be able to put several pieces together in such a way that whatever you are getting, is a valid chemical structure. Luckily, you can actually learn how to do it. You don't need to have the data for these properties. You just can create kind of artificial data where you take pieces of molecules and you learn how to do this completion. And this is the idea behind this approach. Again, the beauty of the approach that for each property what pieces are likely to guide it, and you see how they are assembled together. So as a human, you can actually look at the properties, see if it makes sense to maybe add some other groups that you kind of a firm believer should be helpful. And here we demonstrate whatever in some benchmark data set that it really kind of works pretty well for multi-parameter optimization problems. And for those of you who are interested, you're welcome to look at the paper. So that's pretty much what I wanted to say. Yeah, I'm on time. Okay, thank you.
Thank you, that was great. While everyone is putting their questions in the chat, I would like to start with a kind of more high-level question. So, medicinal chemist friend of mine, he always talks about learning and speaking the language of chemistry. Mm-hmm. Now you have also done a lot of work on natural language processing. Is there synergy between working on chemical structures and natural language processing? Can you take aspects from one field to the other and improve perhaps not only the predictiveness of the methods, but perhaps also the understandability or the interpretability of the methods?
Yeah, thank you. This is a great question. So in natural language processing, it's very interesting. If you think about like how machine learning got into natural language processing, one of the unique features was how do you add the structure and how do you model the structure, syntactic structure of the sentence to obtain better results. So, however, as the neural models become stronger and stronger representation for sentences, kind of syntactic structure faded. And today we have transformer and many other types of representations that sort of indirectly encapsulate the structure of the sentence. What happens in chemistry is quite opposite. There are models which take smiles of the molecule, which is a string, and try to use traditional, string models to represent molecules. And, I firmly believe these models don't work. So based on what I observed in the field, the models that can in a richer way capture the structural features of molecule do better across many different tasks. But this is a matter of discussion. People always have opinions about it. So I think both fields think about how do you go beyond the structures, which are not the strings, but other types, more complex structures, and think how to translate it into algorithms. Also, I started working on interpretability actually in natural language processing, thinking about, rational extraction for text. And the model that I just showed you has a lot to do with, our original model that was producing the extractive rationales with reinforce. So I think the two models are the two areas are quite close, and there is, like, when I'm thinking about them, there are a lot of kind of ideas migrating from one to another. I still didn't really manage to take something from chemistry and to put it in natural language, but there is always a place to innovate.
Great. The first question is by Günter Klambauer, who was wondering whether multitask learning could significantly improve the predictiveness of your models.
Absolutely. So actually the Chemprop, the model that I presented, can be easily trained in the multitask fashion. So you can just, instead of predicting just one property, you can be predicting multiple properties, and it makes sense because when you're screening the library, you're typically screening it across many different properties. These properties often correlate. So absolutely it can help, especially if you have little data for one of the properties.
Next question is whether different conformations of the same molecule can be used as using three-dimensional information from the graph convolution.
So in theory, so this is like a kind of question that always comes back, you know, how do you incorporate three-dimensional information for graph convolution? You know, algorithmically incorporating this information, it is possible indeed. And there is a faculty at MIT, Professor Rafa Gomez-Bambarelli, who very actively works on this topic. Unfortunately, you know, if you look across the board, I think the technology is not at the point that these 3D models are competitive against, you know, the standard kind of 2D view. But I think it's something that's, you know, as we become more sophisticated with methods, it's going to change.
Okay, great. And then you stepped on on a few people's toes by saying that smile-based prediction doesn't work. So there are a few firm believers in the chat that this actually does work. But I guess that would be a coffee break discussion if we had a coffee break. So let me continue with the next question, which is about how much of a performance gain do you actually get from using edge features as opposed to only using the nodes?
So I wouldn't remember in my, you know, the numbers, but in the paper we actually answered this question, and we do see there is an improvement from utilizing edge features across multiple properties. Now, if you look at this Chemprop paper, let me just put the link so that whoever is interested can check it out. One thing that I should definitely mention is that, you know, a lot of these conclusions are kind of specific to properties, because we're thinking of the properties in a very naive way, saying, you know, it's property number one, property number two, but they're clearly very different properties. In some of these properties, it's really important to have the right local patterns. For other properties, it's more important to have kind of the more global pictures, the size and other things. So for majority of these properties, we've seen that the edge, modeling edge actually does make a difference, but not to all. And for numbers, please go ahead to the papers. I wouldn't remember. Sorry.
Thank you. There is one very good, very interesting last question from Hakim Öztürk, who asked whether with all the things that you learned from kind of the graphics representation, can you make a better version of smiles? Can you go back and go to a string representation of things that is somehow capturing chemical essence more meaningfully than smiles does?
So I would first say that on the personal, there are so many people who try to make smiles work that, you know, I wouldn't enter that camp because there are so many already there. So I don't think they need another one. I kind of, to be totally honest, I just don't see the reason why would you do that. Like, you know, like I think that I talk to people who develop original models working on smiles, and at the time, you know, there were a lot of good models in machine learning that enables you to do a great job on smiles because it's a string. But now we have a lot of models that work on graphs. So like, you know, why to constrict yourself and focus on the, you know, on the sequences? To me, the question is, you can do it with a sequence if you love the sequences better. You can do it with a graph if you love graphs better. I think there are a lot of unanswered questions. Like 3D is one of them. The questions about generalization to new chemical spaces, our full inability to use pre-training, to use raw data. So those questions, they are applied, either you do it at the graph level or do you do with smiles. That's where, you know, I would put my personal, you know, time to try to solve these questions. So
thank you. That was